So good morning everyone. Welcome to this lecture. Welcome to another lecture in the series of immunology. Today we see what we'll be talking of today. Till now we have a very clear cut understanding. We established different concepts of immunology from basics. What are the components? What are the different types of cells that are involved? What is humoral? What is acquired in brief? Then the way that these particular cells are being produced, the secondary lymphoid organs, and then we had a very in-depth understanding of humoral, or we should say how B cells are mounting the immune response, how antibodies are being produced, and after producing, what are their functions, what are B cell receptors, how those B cell receptors are activating the machinery or the signaling inside the B cell, and how that B cell is increasing its number, it's getting differentiated into plasma cells and memory cells. So that was a axis of humoral response. Today we'll be starting with another player, another very potent player, that is called as T cell, and we'll be starting today to understand what is actually when we say that yes, T cells are involved in cell mediated immunity. What do you mean by this terminology, cell mediated? Yes, I told you earlier that it's a part of acquired. Both humoral and cell mediated are part of acquired. So today we will be talking about the second part of acquired immunity, and that is called as cell mediated immunity, in which T lymphocytes, the T cells, are responsible for doing the effector functions. Now, how many types of T cells are there? See, we already know two types, T helper and T cytotoxic. T helper and T cytotoxic. So today we will be establishing a clear cut understanding after this lecture that what are the different types of T cells that we are talking about, how they are mounting the immune response, and every aspect will be touching in detail. So let's begin with the general part that when we are talking about the T cell, what are the roles of T cell? What are the roles of T cells? T cell is doing nothing. T cell is doing nothing. T cell is actually not directly coming in contact with the antigen. It needs certain mediators to activate its own self. Now, what I meant by that? What I meant by that? See, whenever there is an antigen, I told you that that antigen will be processed by APCs, antigen presenting cells, and then that antigen which is processed is presented on the surface of the cell by the MHC molecules, by the MHC molecules. Now, when we are talking about, please understand it clearly that when we are talking about T cells, there are different types of T cells, T helper cells and T cytotoxic cells. That's the two major population that we'll be talking about currently, first to have an understanding. And then there are many type of cells like Treg, that is their regulatory T cells, that are again very potent in today's scenario. In today's research scenario, regulatory T cells are one of the most important aspects of immunology. And then we have T17 lymphocytes that we say that they are secreted in terms of your IL-17 secretion. But yes, we'll be going to the advanced section in later stages. First of all, let us understand that we have these two subtypes of T cells. Now, T helper cells, as per the marker, are CD3 positive, CD4 positive, and TC cells, that is cytotoxic T cells, are CD3 positive and CD8 positive. Always remember this nomenclature. If you are a graduate student, then you should remember at least this part, that the marker that can be utilized to differentiate between the two subtypes of T cell is actually CD4 and CD8. The CD4 T helper cells and CD8 are cytotoxic T cells. Now, the another important aspect that you should be able to understand clearly, See, when APCs are processing, APCs are the type of cells which contain both the classes of MSC, MSC2 as well as MHC1, MSC2 as well as MHC1, both. Whereas The remaining types of cells that are present inside the body, all the nucleated cells which contains nucleus, they contain MHC1 molecule. MHC1 molecule. What is the job of MSC2, MSC1? We already understood it in the previous lectures. Now let us recapitulate. MSC2 is involved in processing the external or exogenous antigen, and the presentation with MSC2 leads to the binding of T helper cell. T helper cell will be recognizing the antigen which is presented on MHC2. On MHC2. Whereas the molecules which are presented by MHC1, those are actually recognized by your cytotoxic T cells. So what is happening? When the antigens are processed, they are presented, say, by MSC class 1, then cytotoxic T cells will be coming, they will be binding with the presented antigen along with the MSC, and that is the responsibility of TC cells, which are resting to identify, get in touch with the presented antigen on the surface, and then let's start mounting the responses. Let's start mounting the responses. And this is how, after binding, this cytotoxic T cell will be getting activated. Once activated, once activated, they will again perform their function. They will go and bind to the viral based antigen. They will induce the cytotoxic T cells. Why we are calling as cytotoxic? Because when they bind to the cell which is presenting the antigen with MSC1, they will induce the lysis of the cell. So suppose there is an epithelial cell in lungs. It is infected with the virus, and it is presented the antigen along with the MSC. The antigen, viral antigen that we talk about, is presented here. The cytotoxic T cells will be coming, recognizing it, that yes, this particular cell is actually infected. Now, how they will come into this cell? Before coming, they are always in resting stage. There is a need to let them wake up from their sleep, that please wake up, there is an infection. And that waking up call is provided by the APC's MHC1. APC presenting with the MHC1 will be recognized by the resting cytotoxic T cell, that yes, now the infection has taken place, we are activated, now we have to circulate in the blood, and they will circulate in the blood, and they will go to the tissue which is actually being infected by the pathogen, and where the antigen is presented by the MSC1 molecule, and then they will bind to that particular MSC1 and the antigen to cause the lysis of the infected cell. Please remember always, the cell which is infected and presenting the antigen with MSC1 will be dying. Now that's the part of exogenous antigen. What about endogenous antigen? The antigens which are produced inside our cell, like tumor antigens. A cell is transformed into tumor, then it expresses different kinds of protein, and that those proteins are expressed on the surface. Now, please try to understand. Let me remove this particular thing from everything. There is a cell, suppose it got transformed into cancer cell, and now there are multiple copies. Now, there are multiple copies of this cell which is forming a tumor-like growth. Now, in every cell, what will be happening? There will be an additional new proteins which will be present, and those proteins are termed as oncogenic proteins or oncoproteins or tumor antigens. Those proteins will be expressed on the surface by MHC1 molecule. Now this is MHC1, which is present in all the cell types, so that will be presenting the tumor antigen. Once presented, once this tumor antigen is presented, then again, this particular cytotoxic T cell will be binding, recognizing, and will be responsible for lysis or cell death, inducing death in the immortal cancer cells. And that, my dear students, is the basis of immunotherapy, when we say that yes, we are going for cell mediated immunotherapy, and that these biomes are already explored, are under trials for various cancers and therapeutic regimens. What is the idea? Idea is to identify the antigen, tumor antigen, which is presented on a normal cell, on the cancerous cell type, whether it is lung cancer, liver cancer, pancreatic cancer, they all will be expressing the MSC1 with the endogenous antigen. And then it's the responsibility of the cytotoxic T cell to recognize that antigen and cause the lysis of the cell. So that is the end part of cell-mediated immune response, that how cytotoxic T cells are getting activated. So I told you the complete axis of this particular part, right? But what is this? What about this? This is helper cell, TH cell. Now, how TH cell is getting activated? We had already made a very clear-cut understanding. What? That whenever antigen presenting cells will be presenting the antigen with MSC class 2, MSC class 2, so there will be say macrophages, B cells or dendritic cells that have presented the antigen with the MSC class 2, and then the T cell receptor will be coming and binding to this MSC class 2 and then activating the T helper response. And this T helper response will be secreting various cytokines. What type of cytokines? Like IL-2, IL-2, 5, 6, 4, 10. All these cytokines will be responsible for increasing the population of this type of cell, the clonal expansion of T helper cell, as well as the clonal expansion of B cell also. So we are right now talking about T helper cell. It will increase the population of this particular cell in number, and then the number of this particular helper cell, which actually recognizes a particular type of antigen, will increase. Once it is increased, it will lead to the form. It will lead to the activation of this particular cytotoxic T cells. This particular cytotoxic T cells. Now let us see about actually how it is happening. So we have two subtypes, CD4 and CD8. Now CD4 is activated by phagocytosis, or you can say phagocytes or antigen presenting cells. Then CD8 plus is responsible for killing the cell which is actually infected by the pathogen. So what is the difference? CD4 is actually working towards killing the pathogen or the antigen. This CD8 is actually working to kill the cell, which is infected by the pathogen, So that's the difference. We are targeting antigen also, and we are targeting the cell which is infected by that pathogen also, so that the complete elimination should take place. Then again, cell-mediated immune response can be divided into two parts, like B cell response. It's a primary and secondary response. In primary response, see what is happening. In primary response, I told you the complete phenomena. So first time, whenever the exposure has taken place, the type of TC cells and T helper cells which have been activated, they will be increasing their own number. They will be increasing their own number. And that will take several days. Why? Because right now, first of all, they identified the antigen, and then they differentiated in response to that antigen, and then they divided to form a multiple population. And among that multiple population, there are two types of cells that are formed. One that is effector, that will be responsible for carrying out the main functions, and other are memory T cells. So always remember that the memory is stored in both the forms, in B cells also and in T cells also. There are memory T cells which are present inside our body after the exposure to a certain antigen. So that is the part. Then we have secondary response, when the next time the same serotype of the pathogen is infecting, then this particular memory T cells are already there, then the time lag will be very less, the response will be very high and faster, and then it will be more effective to stop the infection, Now, let's talk about self-mediated immunity in detail. So T cell, what they do, they can only identify the processed antigen. You have to understand that if a complete microbe is entering, they are not able to identify it until and unless this microbe is taken up by the cell, processed, and a small fragment is presented. These T cells will not be able to do any function. They will not be able to do any function. So these T cells are see, these T, because of this particular region, these T cells are very much suited or very much adapted for cell-to-cell interaction, and they target those body cells that are infected by the pathogens directly, like viruses or bacteria, or we are talking about the endogenous antigens. So that is a reason why they are not able to target the blood-borne antigens, or the antigens which are not getting attached to any kind of cells which are present in the viremia, in a bacterium kind of condition, or in viremia kind of condition. So what is happening here? Let us see here. So how these cytotoxic T cells, after binding to the target cell, leads to the lysis of this particular cell. How it is causing the lysis? Now let us see here. Once it is attached, how it will be attaching? It will be attaching by your MSC1 based recognition pathway. MSC1 based recognition pathway. That is understand. Now, once they have attached, what they will do? This is the cell that we are talking about. This is the infected cell. This is the infected cell's nucleus, and that this lining is the membrane. So once the cytotoxic T cell is attached to the membrane, it starts releasing various molecules in the form of granules, like granzymes. These are the principal molecules, and then perforins. There are two molecules that are specific for this particular cytotoxic T cells, called as perforins and granzyme. What is their role? See, granzyme is the word derived from enzyme. Now the granules which are performing the enzymatic activity are called as granzyme. So how it is done? These enzymes can go and cause degradation of the cellular components. They cause degradation of the cellular proteins and complete metabolic machinery. But they need to enter inside the target cell. How they enter? They entered by perforins. Now these perforins are these perforins are proteins which will go and bind to the membrane of the infected cell, and multiple units of perforins will be opening up the membrane, will open up the membrane. Now they will form the pores that are required on the membrane in order to transfer the granzyme inside the cell. And then once the granzyme enters inside the cell, it can lead to lysis of the cell. And that is how these cells, these cells actually are causing the death of infected cell or tumor cells that we are talking about. This is the real-time image of cytotoxic T cell and cancer cell. So what it does, it attaches to the membrane, and then it releases the perforin molecules. They will create the pore and transfer the granzyme inside it. So that is the thing that you should understand, that how cytotoxic T cells are actually causing the lysis of the cell. So moving ahead, when we had discussed about the lymphocytes in bone marrow, then we clearly established that the lymphoid progenitor that is generated from the hematopoietic stem cell is responsible for formation of these lymphocytes. And among them, after lymphoid progenitor, the T lymphocyte progenitor will lead to the formation of T cells. Now T cells, once they are formed, lymphocytes are formed in bone marrow from hematopoietic stem cell, and they mature in thymus. And after adulthood, they perform their maturation in the secondary lymphoid organ. Then in terms of maturation, what we understand? Initially, all the T lymphocytes are CD3. After being matured, they convert themselves into CD3 and 4, whereas 3 and 8. So that this separation of two subtypes is happening inside the thymus at the stage of maturation. Now, CD4 T helper cells, how it is activated? It is activated with only three type of cells called as APCs, or antigen presenting cells. Now these antigen presenting cells are macrophages, B cells, and dendritic cells. All these three cell types can activate T helper cells. Whereas CD3, which is actually getting activated with the help of MSC1 molecule, can be activated by the APCs, all the three that we have discussed here, MBD, and all the body cells that are present inside the body. That is all the nucleated cells, all the nucleated cells. MSC1 is not present in RBCs. It is not present in red blood cells because these are not having the nucleus. Okay, once they are activated, they will differentiate into effector with cells which will be responsible for carrying out their function, and they will be converting into memory cells. So the memory cells CD4 and CD8 will always be there, but you should understand that after maturation, when we are talking about the activation and exposure to this particular antigen, this is actually happening inside the lymph node. And once in this exposure is happening, what is there? A single cell is getting exposed, and it is leading to the formation of multiple multiple number of T cells of same type. So a single cell will lead to clonal expansion, and that clonal expansion will be happening inside the lymph node. Now, of course, this clonal expansion will be triggered by release and taken up of certain cytokines, and the main cytokine, the principal cytokine, which leads to the clonal propagation of T cells is IL-2, and this is in turn secreted by the activated cell, and by autocrine signaling, it takes up back all the IL-2, and then it starts clonal expansion. Once the number is increased, now they have to reach to the site of infection, and then they enter inside the blood plasma to perform their function, and that is the complete chronology of these cells. That is the complete chronology of these cells. So once we had, till now we have a very clear understanding of cytotoxic T cells, which are actually causing the lysis of the cell. So what is H? What is the function of H? Why we are calling it as helper cell? We are calling it as helper because it will be helping. How it is helping? How it is helping? See, this is a pictorial representation of dendritic cell. It has presented the antigen with the MSC class 2 molecule, and T helper cells have recognized this particular receptor with the T cell receptor, and the co-receptor is also activated. Now, once this T helper cell is activated, fine, the simultaneous process antigen of MSC class 1 is activating cytotoxic T cell. So a single APC can activate both the cells by two different MSCs, that is understand. Now, this activated T helper cell will stimulate this particular dendritic cell. It will stimulate the dendritic cell, fine, and this particular dendritic cell, after getting stimulated with the helper cell, will initiate the binding of cytotoxic T cell. So please remember, until and unless the signal is provided by the T helper cell, dendritic cell will never be able to mount and activate the cytotoxic T cell. And that is the reason why we call helper H cell as T helper cell. So T helper cell is performing the helping function in both humoral as well as cell-mediated immunity. In cell-mediated immunity, it is responsible for activation of cytotoxic T cell, and in humoral, it is responsible for the activation and clonal expansion of B cells. And that is the reason why we use dendritic cell as dendritic cell therapy, because they are very potent cells. They can activate our cell-mediated immune responses also, and by both the actions, it can lead to the elimination of tumor antigen. Why? Because cell-mediated immune response and cytotoxic T cell is actively involved in removing the oncogenic cells, or we should say the oncocells. So that is in nutshell that how these things are happening. Just to summarize you, that T helper cells, once they are bound, then you can see here in present condition, there is no MSC1 that is being secreted or presented. So now, right now, the T helper cell is binding to the MSC class 2, and then it what will happening? It will be increasing its own population. It needs to increase its own population in order to bind to other APCs also. And this clonal expansion is multiple copies of this type of cell which is specific to this antigen. Now all these cells, the morphology of this T cell receptor is actually specific for this particular antigen only. And that is why we say that yes, these cells will eventually develop into T helper memory cells, and they will be responsible for your long-term immunity. They will be responsible for their long-term immunity. Now, once binding has taken place, I told you that they help in humoral immunity. Now, when B cells, suppose when B cells are expressed, I told you that T cells will release multiple cytokines in order to increase the population of B cells. So this is just to recapitulate what we had discussed at the last time. Fine. Now, when we talk about T cell activation, see, we discussed B cell activation. There we studied that there are two signals that are required, first signal and then second signal. Similarly, here also the double recognition is required. The double recognition is required. Now, what do you mean by double recognition? First of all, my T cells should be able to distinguish clearly between the outside antigen that has come to me and the proteins that are part of my body. It should not mount a response towards my antigen. It should be able to identify clearly what is self and what is non-self. Non-self is nothing but outside the body. Fine. Then, in order to identify what they have done, see, one first recognition is always with the MHC. Now, what if MHC1 has presented my own protein on the surface? If this property is not there, then T cell will come and will lyse, cause the death in my normal cells, and I will see my cells are rupturing. So that should not happen. So for that, there is another receptor that works simultaneously, and that process is called as co-stimulation. That even if the T cells are binding to this particular part, there is an additional receptor that is required to activate this, and this co-stimulation will only be activated in case of non-self antigen and never in case of self antigen. That is the way we go for recognition. That is the way we go for recognition. Now let us see that how it leads to activation or co-stimulation, or perhaps we will be talking about that particular aspect when we're talking about T cell receptor and how those receptors are causing that particular proliferation. So once activation has taken place, the T cells, suppose this T cell is activated. Now what is the job? It will go and increase its size. It will differentiate and then it will divide. Then these cells will divide into multiple copies. These cells will divide into multiple copies and they will start producing its clone. So this is called as clonal expansion. And that this complete process is happening where? This is happening in secondary lymphoid organ, and inside that secondary lymphoid organ it is happening in paracortical region of secondary lymphoid, where the T cell center was present, once they are activated, this access is happening simultaneously. Along with this, what they do, these will be starting secreting the cytokines which will start inducing the inflammation, at the site of infection. All the inflammatory responses that we studied, that the dilation should be there, the increase in the permeability should be there. So all those mechanisms, all those cytokines that are required for that particular process will be released simultaneously, so that the antigen is busy fighting the inflammation, and meanwhile we are increasing our army in terms of increasing the number of cells. And that is how the complete process is taking place. Now if you take out the section and see the paracortical region inside the secondary lymphoid organ, you will see clearly that yes, the cells that are present are increasing number, and the activation of lymphocyte is already there. You can clearly observe the activation lymphocyte. In fact, you can use flow cytometry also to clearly see whether the proliferation is happening or not. What you can do, there is a fluorescence dye called as CFSC. You can label the T lymphocytes with the CFSC and can see whether there is an increase in the number, increase in the DNA content. Because anyhow, if this cell has to divide into two, you will see that this one is diploid, tetraploid, and it will be leading to the diploid cells. And that is how the cells are dividing by cell cycle, and you can easily monitor that by using flow cytometry, which we'll be studying, and we'll be talking about how to use this flow cytometry in immunoproliferation, immunophenotyping in the later lectures. So that is in nutshell the complete part of this particular T cell. Now moving ahead and going further in depth, we note cytotoxic T cells. These are CD8 positive cells. I told you we have helper cells that are CD4. Among T helper cells, there are two types of responses based on function. We call it as Th1 and Th2 response. We call it as T helper 1 and Th2 response. Now, why these two categories? Because they perform different functions and they involve different set of cytokines that we'll be discussing. Now, first of all, please be clear. All the three type of functions effect that is given by T cells are against the antigens. They are activated only and only by antigen presentation, and they are performing different functions: lysis of the cell, activation of humoral response, activation of cytotoxic T cell, and there are different things. So let us see. CD8 cells, cytotoxic T cells, will cause the lysis of the infected cell. They will be activated by MSC1. So they are specific for the antigens or the pathogens which can be presented by MSC1, and those are always intracellular pathogens, bacteria which can enter inside the cell, like Mycobacterium tuberculosis or all the viruses that are entering inside the cell. So for those, the cytotoxic T cell is the uh, prime enemy or prime helper for us. And apart from this, you can also write oncocells or cancer cells also. Then we have Th1 response. Now again, it is activated by MC2, there is no doubt, but it is more specific in response to bacteria and parasites that we are talking about, bacteria and parasites. Now, what is the difference? If macrophages are presenting the cells with MSC2, then it will lead to Th1 response. And what they do, they will activate all the macrophages. They will activate the cytotoxic T lymphocytes. So what is there? Th1 response involves the activation of macrophages. Please phagocytose the antigen. They will activate the CD8 positive CTLs. They will activate the CD8 positive TC cells, and also they will induce the B cells. They will activate the humoral response also. Then Th2, it is against the bacteria, that is extracellular bacteria. We're not talking about any intracellular part, and the toxins or the peptides or the allergens that we're talking about. The whole sole motto of Th2 is to activate the B cell or activate the humoral response. So you see clearly, T helper cell, if they are involved in activating CD8, they are called as Th1 response. If they are involved in activating B cells, they are called as Th2 response, and that is how you should remember it always. Now, we can go ahead with multiple scenarios in this particular section. That, like, what this is again a summary chart for you, that what we have read till now. What we have read, we have an antigen. It can enter and can initiate the response. So first of all, your innate immune response will be activated. the surface barriers will be activated, all the defense mechanism. Once it is entered inside the body, then your adaptive immune response is activated. The antigen is taken up by the antigen presenting cells, and these antigen presenting cells will be responsible for activating the T helper cells, and will also be activating the T cytotoxic T cells. Fine. Simultaneously, what they are doing, the activated antigen is also activating the humoral response. Also activating the humoral response. And at the last, what you will see, you will be having multiple antibodies that are ready to fight. You will be having the T helper cell that is ready to activate again this section and this section, and you have the cytotoxic T cell that is ready to lyse the infected cell. And then what you will see, that this antibody will go and remove this particular antigen. The cytotoxic T cell will go and lyse the infected cell, and T helper cells are channelizing both the paths. And this is how the complete summary chart of immune response is there, that what is happening inside our body. So for your understanding, these are certain MCQs that you should remember, certain MCQs that you should have a very tight grip on. So thank you so much for your patience listening, and wish you a very happy learning. We'll again meet with another set of lecture. Till then, please take care and wish you a very happy and safe learning. So thank you so much.